and welcome to Marvel. Marvel is a project funded by the European Commission within the Horizon 2020 FET-PRO-ACT group. Marvel partners are from 5 European countries. The project is coordinated by CNR, the National Research Council of Italy. There are 2 clinical partners, Cardiocentro Ticino from Switzerland and Università Vita e Salute from Italy, and 3 SMEs, Hyperdrop Diagnostics from Spain, Hansa Biomed from Estonia, and Amires from Czech Republic. Marvel mission is to develop the first affinity-based, scalable and reversible isolation technique for small extracellular vesicles, opening entirely new scenarios in both therapy and diagnostics. Indeed, Marvel technology will be validated in 2 important clinical contexts that are the diagnosis of bladder cancer by liquid biopsy and cardiac regeneration by cell-free therapy based on extracellular vesicles. EVs are an heterogeneous family of submicron membrane vesicles released by most cells in basically any biological fluids with a fundamental role in cell-to-cell communication. Much interest is flourishing towards their exploitation for innovative methods in medicine. To date, the highest clinical relevance is thought to reside in exosomes, nanovesicles smaller than 200 nanometers.
Exosomes represent the 3rd-generation cell product to be used in cardioprotective and cardio-regenerative medicine. After the initial excitement over cell transplantation using adult progenitor cells for its relatively easy to use and good safety profile, the initial mechanistic hypothesis has been questioned. To date, we know that cells have been effective by indirect mechanisms, by reducing apoptosis in cardiomyocytes, stimulating angiogenesis, or modulating the inflammatory reaction. Thus, the trophic secreted molecules and secreted vesicles are more critical than the differentiation of cells in the new cardiomyocytes. Protocol to isolate in a large scale the well-defined and most effective fraction of exosomes, as it is for one of the Marvel project milestone, is of paramount importance to move on toward the clinical application of cell-free approach in cardioprotection.
On the other hand, extracellular vesicles have a role in pathological processes as well. Secreted by malignant cells, they have been implicated in disease development and progression by stimulating tumor cell proliferation, angiogenesis, extracellular matrix degradation, and immune response deregulation. Since one of the most important challenges about cancer is the diagnosis at early stages and proper treatment, the identification of biomarkers represents an urgent clinical need. Resembling malignant cells of origin, tumor-derived extracellular vesicles are suitable surrogates to be considered as a source of diagnostic biomarkers. Upon isolation from liquid biopsies, the analysis contributes to implement non-invasive, affordable monitoring of cancer progression.
Marvel key technology can be declined in one simple word: peptides. Our vision is indeed to introduce a paradigm shift in the affinity capturing of extracellular vesicles and use synthetic peptides to replace antibodies.
Peptides' properties can indeed be finely tailored in terms of selectivity and affinity of EV recognition. We will indeed develop different peptide probes acting both as general EV binders by targeting EV membranes and peptides targeting specific EV protein markers to enrich predefined EV subpopulations. We will take advantage of the versatility of peptides as synthetic molecules to enable their conjugation to different supports. Conveniently, we will also design and apply different strategies to enable the release of extracellular vesicles under mild condition, preserving their integrity. We will then exploit these concepts in the integration of our affinity systems across different settings and operating scales, including LFTs, laboratory kits, and TFF membranes.
In Marvel, the breakthrough is to develop an affinity membrane unit for the large-scale purification of extracellular vesicles by using highly selective hybrid probes specific for subpopulation of the vesicles. Currently, for the isolation of vesicles is applied sequential filtration by integrating different membrane processes, microfiltration, ultrafiltration, diafiltration, but one of the drawbacks of the site-based isolation is the presence of nanoparticles with the same size of the vesicles and the presence of contaminant molecules. In Marvel, we will develop an hybrid of modified membrane unit to be integrated with the tangential filtration system that currently is used by enhancing efficiency and reducing contaminants.
Over the last few decades, EVs have become sort of magical bullets that fueled high expectations for new generation diagnostics, omics, and therapy. Hansa Biomed has pursued this long-term vision since 2007, becoming a global pioneer in providing EV-focused solutions to life science market. While today, along with Marvel Consortium, we're looking upon other markets such as that of precision diagnostics and regenerative medicine, which impact reaches millions, billions of euros in high-interest hubs such as Europe and States, and can bring benefit to hundreds of millions of people worldwide. We also consider other markets such as nutraceuticals, cosmetics, and consumer care as early release markets that are even more lucrative with less regulatory hurdles and shorter product development cycles.
For paper-door diagnostics, as a rapid diagnostic test startup, Marvel will provide novel and disruptive know-how that will be applied in new products and future collaborations. From our experience, European projects have always helped to increase the synergy between research groups, industry, and healthcare systems. Technologically based startups can benefit from European innovation programs to improve the technology transfer flow from research and industry to the European healthcare system. Thanks to the funding obtained, startups can hire highly trained personnel, such as PhDs, and explore cutting-edge and multidisciplinary projects.
In 2020, EV therapeuticals have entered human testing all the way to COVID-19 therapeutical trials, but developers still face some challenges regarding the EV exceeding heterogeneity and complexity that impact GMP, regulatory QC issues. So within Marvel, we want to reshape the state of art and advance it versus truly affordable, scalable, robust solutions that we will then launch either within existing pipelines or we will establish strategic partnerships with therapy developers or dedicated startups. In any way, we will try to attract new public and private funding.
Marvel will address different target groups, including research community, industry, SMEs, end users, broad public, policymakers, and European Commission. The website and social media will be one of the main windows into all our activities and results. We plan to directly engage the broad public not only through articles, press releases, non-scientific publications that we plan to publish within the Marvel project, but also through organization of different events that we plan to organize over the next 2 years. And finally, Marvel project will also engage into certain clustering events with the participation of policymakers and European Commission, where we will present all the possibilities of the Marvel project to those target groups.